Yeah. Yeah. Yeah, that's what I'm doing.
No, it's still time. Okay. Good, why don't we leave the door open? I know something the others will come in. Yeah, leave it open for now. Oh. Yeah, I know. You even have a matching t-shirt on. Oh, the speaker says that.
If you would like one, I will make the plan.
She did, nice. With the color on, I'm sure many people like it.
Let me do this and you get the full effect.
We don't wait one more minute, because the online people,
Still joining in. Will I see if they do anything?
Huh?
Will I see any reactions from them?
We will check later.
Okay.
Yeah, we will check later.
All right.
But usually not. For our students, we have students who are in Nigeria and Uganda and Mozambique, and they usually listen, like we try to. So, yeah, so it's mainly us. Oh, no, I haven't seen you. Or you're doing this course. Yeah, I can take it. No, I'm sorry. Oh, you're doing it in the back. Just doing it through the whole. I know, it's a test. I know, I thought you were going to make a practice course. I know, well, that's amazing. Sorry. Yeah. Yes. Yes. Hello, everybody, and some new faces. We have new honors students starting today.
That's fantastic.
Yeah, so it gives me pleasure to introduce them. Some of you will know him well because we work together on a joint paper. But we first put your PhD. So you did a PhD at the IBM and joined from background labs. Then you worked at a postdoctoral research position in KwaZulu-Natal, researching for TB and HIV, isn't it? And then you came back, and it has been at the CPGR. He will tell you more about that for 5 years. And 2 years ago, there was a merger between the procurement work at the IDM and the CPGR, and this is now 1 year. It becomes really complicated, so it's DCY. So that's now the new unit name. So that's the one you about. And you will discover very quickly that human is very efficient for the protein. And I think it's really wonderful that the opportunity to get an update what the unit is doing, and it's an interesting title. What is proteomic and is it worth my time? I'm sure it is. Thank you for making the time to speak here. Thanks.
No problem. Thank you, everybody. So my name is Liam. I'm happy to field questions as we go along if you've got any. Or else we can do it at the end. It really doesn't bother me. And the test, I think you said, was afterwards. Warren, you said downstairs we were writing the test. Okay, all right, great. All right, so just to give you a bit of I'm going to walk you through this. So talk to you about the Decipher origin, a bit of Bio 101, show and tell, strictly PG. Talk about how small things can have big impacts, specifically proteins within your body. Why are proteins important? What would be your typical diagnostic odyssey that clinicians might have traditionally followed and where they might go moving in the future? Moving on to study design, what's sort of an old school study design and what the new school study designs are, how have advances in technology allowed these newer things to occur, and then I'm going to run through a couple of different literature sort of articles that are out there and how they've been applied. So Decipher, if you look up a few industry or dictionary definitions of Decipher, you know, a lot of the words understanding, discovering, determining meaning, making things understandable. I'll leave you to read those in your own time, but that's the purpose of what Decipher is trying to achieve. So a lot of people tend to think of Decipher as some kind of acronym and figure it out. It's not an acronym, it's a purpose, right? So this is what we're trying to help you do, is decipher what's going on. In terms of our origin story, the DSI has funded different entities like the NRF, et cetera. Some of those entities are the Technology Innovation Agency. Some are the South African Research Infrastructure Roadmap, which is SARIR. SARIR has a number of programs within it, one of which is Diplomix. So Diplomix is specifically, it stands for Distributed Platforms in Omics. It's specifically focused on omics technologies. TIA funds the CPGR, which is the Center for Proteomic and Genomic Research. And then Diplomix has enabled both the IDM and CPGR to merge their platform, their proteomics platforms into Decipher. So there are a number of programs or initiatives from Diplomix. You'll see at the bottom here, there's Decipher. Clarity is a sort of a marketplace for bioinformatics. So think of it as Airbnb for bioinformaticians. If you do bioinformatics and you're looking for a job, you can go to Clarity. If you're looking for a bioinformatician to do a job for you, you can go to Clarity. INGWE is a rare diseases initiative that's looking at using multi-omics to try and help people in the rare disease space. And One KSA is connected to the biogenomes projects that are happening around the world. And in particular, the focus for this one is to do 1,000 biogenomes within South Africa. Okay, so Bio 101, I think most of you will know that, you know, you start off with DNA, DNA then becomes RNA, RNA translates itself into an amino acid chain, which then folds itself up into a protein. One thing I want you to take away from all of this is that your DNA is static, right? Your DNA doesn't change because you ate something 5 minutes ago. Your DNA doesn't change because you've run up a hill. Your DNA doesn't really change much, full stop, right? Whereas proteins are changing all the time in response to infections, response to what's going on around you, stress, everything, okay? So where that becomes important is in the context of trying to study phenotypes of what's happening. I think that proteins would be a more, probably you get more bang for your buck by going that route. I suggest you guys scan this code. and then go and watch this on YouTube. This is a video that is a 3D video, placing you inside a cell. Come on in, take a seat. Placing you inside a cell. When you move around, if you can use it with a VR headset, you literally can look all around. I am many decades old, and I saw this video in February, and it left me speechless. And it taught me things about cell biology that just made absolute sense. The points I'm trying to make will be very informative. So I highly recommend you go and take a look at this video. From within this video, just to point out that this is actually screenshots from within the video. This is your nucleus right down here. And from within this nucleus, what happens is you, obviously, if your genome gets messed with, then you have serious complications that occur. To protect it from that happening, it's placed inside the nucleus. And there's then a mechanism for getting from DNA to RNA, which then creates your proteins, right? So what you're seeing here is the ribosomes that are attaching to a strand of RNA. And this then subsequently goes and attaches to the endoplasmic reticulum. And as this chain feeds through, the amino acid chain is being formed on the inside of the endoplasmic reticulum. And what happens then is it'll then go through your to your Golgi body, get packaged up, and your Golgi body is like a bit of a post office, right? It says, okay, this protein needs to go there, and it'll package it up, put it into an exosome, and send it off to wherever it needs to go. From within inside the cell, if you look right up at the top here, you can see that's actually your actin cytoskeleton of your cell, right? You might recognize mitochondria, powerhouse of the cell, what's going on there. And there's any number of things. The point that I want to make with this is that your cell is so much more than just DNA. There's a huge number of molecules that exist inside there that can tell you information about what's going on. And if you had to take a look at this as a comparison, if you compared just volumetrically as a direct comparison of how many proteins could exist in your cell, just the variance of what could be there versus a transcriptome, as a volume of water comparing the pool to the bath. Like that's the difference in the possibility of information you could be looking at. And then we take that one step further, right? So a cell is a cell, but then all your cells conglomerate together. They form tissues. Tissues form organs. Organs are connected to each other, speaking to each other. So your body as a system is made up of all of these functional parts that are constantly communicating to each other. Once again, going back to that dynamic protein signature of what's happening in the communication versus the very static genome side of things. So from a clinical aspect, proteins are incredibly important, right? So this is from, you can look at the paper here, but basically the Institute for Laboratory Medicine at the University Hospital in Munich, they looked at inpatient and outpatient, but specifically at inpatient. 9 million inpatient tests were run in 2016 in one hospital, okay? Of those, this is kind of the breakdown between, so you're looking at the blue across these two. So this is inpatient and outpatient. Of those, over 40% of requested tests were protein-based. So from a clinical perspective, diagnostic decisions are being made based on proteins. And they're being made based on proteins more often than any other class of molecule. So from a research perspective, we don't just get to a diagnostic test. We first have to develop and go all the way back. So what is that diagnostic odyssey? So many times what's happened in the past is you might say, okay, I know that there could be potentially this problem. I'm going to test using these 3 variables. Based on the outcomes from that, I will know, okay, it's this or it's that, and I can make a treatment decision. That's sort of clinicians, not a clinician, but when I talk to clinicians, this is ultimately what tends to happen the majority of the time. What we don't want to do is develop new biomarkers that where we don't, it's just, you're splurging out data. Here's just a whole bunch of things, go and take a look at it. But it's giving you no context, it's giving you nothing additional. So whilst there is extra information, without the context to it, it's useless information. What we see happening in the future is that you're going to be able to profile a whole bunch of different proteins. You're going to look at your patient history, the clinical data, all of these things, and you're going to have algorithms that are going to help you. And you saw this with the panels, right? Not a single point that's telling you a piece of information, but it's a panel based on multiple things that are coming back and giving you a feedback on what could be your clinical diagnosis or what should you be doing as your next treatment step. Okay, so just a recap, managed to get through those things, and we'll be moving on to the next. In terms of biomarkers, a lot of you might come across the word biomarker. A lot of you might be thinking about doing studies that involve biomarkers, right? Just a really quick recap that they can be any kind of class of molecule, nucleic acids, small molecules, proteins. But what they should definitely be is quick, robust, and cost-effective. Those are having a really good biomarker, but it's so expensive, it's only available for 1% of the world. It's not particularly useful. Likewise, having a biomarker, but it's really inconsistent, is not useful, because when a clinician uses it, they say, but maybe it could be this, maybe it could be that, because I know that this biomarker could be inconsistent. And obviously quick in terms of when you're making diagnostic decisions, you'd like that feedback as quickly as possible. So when looking for new biomarkers, you can go and read this, this textbook that was produced about 5-6 years ago on the best kind of process to get towards new biomarkers. And in particular, I'm going to be talking through most of this. This tends to become, once things go into a clinical validation point, and this tends to be preclinical when you're testing and then validating what you're finding. So when you are designing your experiments, really please make sure that you're not just thinking about finding something, you're thinking about how do I validate what I found as well. And then another useful concept that I find in terms of experiments is you divide them into 3 blocks, right? There's the pre-analytical part of your experiment, there is the analytical part of your experiment, and there is the post-analytical part of the experiment. And things in all of them are connected, right? So pre-analytical is sample collection, storage, transportation, extraction, all of these things. If you are inconsistent with that, you will be inconsistent with your endpoint. So you really need to make sure that if you are, for example, doing a urine collection for your project, that urine is consistently collected in the right way, right? Not some are sitting on the bench for 2 hours, some are processed straight away, etc., etc. That needs to be clearly defined. Storage is consistent, it's monitored, all of those types of things. And then likewise, if you, on the analytical side, which is where we would focus, is making sure that we're consistent in our preparation of those samples. We're doing things like system suitability tests. So how do we test our machines and make sure that they're performing and that they're not highly variable? Because what's the point in trying to measure points of data when what you're using to measure them is all over the show? And then in terms of the post-analytical, you need to be able to QC this set of data that's come out there. You need to be able to interpret it, analyze it, and then potentially take that forward for validation. So these are all things that you need to be, from your experimental side, need to be thinking about. So with that in mind, old school designs have tended to focus on this triangular design, where you would start off at a discovery phase, you'd focus on a small number of people or subjects or whatever the case may be, but you'd be looking for as much as you can. And then from there, you'd kind of say, okay, we think we've identified what we're looking for, or a list of things that we're looking for. We're going to now verify that using slightly more people and a smaller target list of what we're going after. And then when you validate that, you'd say, okay, now we're really going to blow the lights out in terms of how many people we test this with, but we're going to focus on a very small set of things. And that would typically then involve, you do something like proteomics up front. You'd then move it maybe onto a targeted proteomics assay, and then you'd maybe move it onto an immunoassay to kind of work your way through. really think that this new school way of going about things is going to be a much more effective way of making things happen. And this is more a rectangular sort of study design. So if you think about being able to take a large number of people, and this is borne out by, if we go this route, oftentimes what happens is costs for doing this initial bit have been really high, right? But these costs have dropped dramatically. So why not get as much information as early as possible, as consistently as possible? So with that in mind, what you can start to do is design these experiments where you have a large cohort, you do your analysis, and what you might find is that there's a whole set of proteins that you identify. This green blob over here might be some that are significantly changing for this particular cohort. And then what happens is when you repeat this experiment on a validation cohort, so a completely new cohort, but with similar parameters, but you're not using the same people fundamentally, you will identify a whole set of proteins. These ones will be significantly associated with this cohort. But what will happen is you'll get this overlap of things that are regulated and significant for both. And that's a really fundamentally powerful way to identify what is your starting point to look at. And that by doing that, you can look at not only your diseases, but you can look at the risks associated with them, the treatments that come with them, and take into account people's lifestyles as well. So if you had to think about all of these contributing some part of a puzzle that you're putting together, you then use all of that to be able to understand what's going on. So in terms of the technology, 5 years ago, we were doing fast-based sample preparation. A skilled technician, like some people in the room, would take a day to prepare 24 to 30 samples. You'd be guided by how big your centrifuge was that you could put tubes in. You'd then run that at sort of 10 to 20 samples a day on your mass spec. High overhead. So what happens is your mass spec acquires data, but if you have it from you inject a sample and then you inject another sample, if that is a 90-minute period, you're actually only acquiring useful data for about 60 to 70% of the time. The remaining sort of 30 to 40% of the time is what we call an overhead. It's preparing the system for the next injection. So fundamentally not very efficient. And then acquiring data in such a manner so that there's a lot of missing values, which makes it difficult. So if you think about cohort size studies, you know, possibility for inconsistency in sample preparation, difficulties there, not really quick in terms of throughput, and you know you've got this problem. There's this problem in terms of missing values. What's happening now is you can do automated sample preparation. You can include a bunch of different kind of kit reagents that are fundamentally important in terms of QCs, like for data analysis afterwards. There's new instrumentation. Incidentally, we have all of this in this pipeline, by the way, as well, which allow you to do anything between 60 and 500 samples in a day in terms of your data acquisition. Processing that is able to deal with this effectively, that you don't have to put on a cluster and it takes you 5 weeks to just try and get your data from a raw format to an Excel file. And then an ability to then go and interrogate what is the most important, intimidating thing about omics. You get any omics data back, it's just this list of stuff and you go, I don't know what to do with this. So how can we help you sort of go from that primary data analysis that turns a raw data file into a list of things and into your secondary data analysis where you go from your list into something that might be sort of a representative figure 1, 2, and 3 in any given sort of paper that you have. And so how can that be done in an effective way if you don't know R or Python or any of these things? And then with all of this, you know, how do we also, we've been able to improve in terms of missing values. So what this leads sort of as a sum of all of this is means that we're far better positioned now to do clinical based cohorts than we have been at any point in time prior to this. Just another recap of where we've got through. So a whirlwind tour up until now, and now I'm going to talk you through some examples of where this has paid off for others. Okay, so we'll start off with, has anybody heard of Genomics England? You have? Okay. So Genomics England, they basically sequence full genome sequences on 500,000 odd people in England. Okay. they then, a consortium of pharma companies then went and said, okay, let's take a subset of that. So they took 50,000 odd samples, 54,000, and they ran Olink, which is a specific type of technology, but it's a proteomics-based assessment of these samples. And from that, they were able to look at quantitative loci, quantitative trait loci. So if you think about West studies and things like that, what those have looked at in the past is maybe where you have a particular gene and where that gene might be responsible for certain phenotypes that you are seeing, right? So what this has done is looked at where you have cis and trans pQTLs. So cis is for local regulation of proteins, and trans is it'll impact proteins across your whole genome. And this here is a measure of count, right? So some of these things are really, really high. Some of them are really, really low. So they basically reported that there were over 14,000 genetic associations with differing protein expression levels. 81% of those were novel. So had they only looked at the genomes, they would have missed 81% of important data, where there were completely novel things based on protein expression. So if they just looked at that one set of information, they would have, all of this would have been thrown out. So it just shows the importance of working in a multi-omic space. I mean, I am talking about proteomics, but you don't necessarily, there is a place and time to do things like genomics, transcriptomics, all these things. I'm just saying, don't forget about proteomics. And so the chief scientist at the UK Biobank said, something along the lines of being able to do this led to more, the data being more than the sum of its parts. And that, had they only done the proteomics, they wouldn't have been able to trace it back to the genetic associations. And had they only done the genetic associations, they would have missed these novel things that were happening. So next example, dose response curves. So we all take all kinds of medication, Panado for this, ibuprofen for that, whatever the case may be. Different people will take different doses, and you will see different responses. One person might respond to one Panado, one person might respond to 5 Panados. It just is dependent on a number of things, right? So what this research group did is they took 13 cell lines. They took 6 classes of compounds covering 31 different drugs. And then they looked at if you had sort of different concentrations, what was the potency effect of these drugs on these cell lines. Okay. This is also a QR code. So if you want to scan this, you can go and use this tool. It's called Decrypt-M. I'll talk to you now about where that's really interesting. So they looked at fold changes of different proteins, and then they looked at fold changes of or the potency that was of those fold changes. So something might go up, but it might have a small impact. Right? So it goes up 3 times, but maybe there's a negative feedback loop or something that normalizes that. So the size of that effect might not be as big. And sometimes things might go up a little bit, but have a big impact. So looking at the potency of what those effects are was just another way of showing that the data is not just about how much dose is there, but what is the impact of that dose. And then they also showed that on 4 replicates that they were within half a log of what that was in terms of reproducibility. So what that fundamentally looked at was they looked at drugs are targeting certain proteins. Those proteins have certain impacts within the cell. And then if you look at, based on their kinase substrates, what that then means. So it's looking at the target, looking at the impact within the cell, looking at the substrates of what these targets are. And you can see that the impacts are very, very different across the board. So once again, looking at how dosages can impact what's going on is fundamentally, if you think about this from a pharma company perspective, It's going to be incredibly useful information. So what they did was they took 10 kinase inhibitors, and then they looked, this is the number of phosphosites that they were hitting. You can see, so some of them, and this is the potency with which they're doing it. So some of them are just broadly hitting things across the board, and some of them are quite refined in what they're hitting. If you have to take this information and overlay it on certain pathways, or MAP kinase pathways, or mTOR pathways, all of these kinds of things, you can see that there are big effects that are happening. And sometimes there are effects that are happening off target, right? Like, so you might be thinking that your drug's only hitting one pathway, but it could be hitting others as well. So the interesting thing that comes out of this, if I had to summarize that, was they took 13 cell lines. They tested it against 6 classes of compounds. The dose response curves were analyzed using Decrypt-M. They produced the software now. And that's unearthed tools to monitor at a pathway level these responses. And the ultimate impact of that is there are off-target effects that are happening. And can we repurpose drugs? Do you know how long it takes to put a drug through clinical development? It's a huge amount of time. And we have a whole bunch of drugs that are sitting there that could potentially just go, oh, you're using it for the wrong thing. Use it for this. Or it is effective there, but it's also really effective here. In terms of proteomics, we sit with a bit of a conundrum. Okay. How do we take proteomics from something like this into something like this? So these next set of slides are from Jacob Warwinkel, who was at the Ubuntu Proteomics Summer School this year. And I want to speak about, you know, if you think about those 2 blocks that from that previous slide, we're focusing here, which is that block on the left, as opposed to here, which is that block on the right. This here was a COVID testing facility that was set up, right? All very standardized, all like sample comes in one point and just a factory to process and output data on the other. So how do we take proteomics from how it's more often than not currently implemented into being something that can be utilized like this? Okay, so if we look at the playbook that a company like Biognosys uses, they're a CRO, so that stands for Contract Research Organization. And what they do is they are contracted by pharma companies to go and do research at a proteomics level on finding off-drug effects and a couple of other things. I'll speak through one of their examples. But basically what they do is they will start off with a discovery-based, so this is your first set, right? Discovery-based, try and identify a number of things, validate that, and then more often than not, they'll move that into a slightly more targeted, it's called a PRM. So it's just where you have a list of targets using a mass spec that specifically is focused on those. And the reason why you do that is from in a clinical environment to get it validated and put it through all the regulations. That's the simplest way to get that done. So this is one of the studies that they've been involved in. The only reason these slides exist is because this was actually eventually published. But most often, these kinds of studies go under the radar. It's private IP. The pharma companies are holding on to this kind of thing. So targeted protein degradation is quite an interesting process from a therapeutic drug design. The idea here is that your body inherently possesses mechanisms through which to get rid of things, right? And one of those mechanisms is to tag proteins with a ubiquitin tag. As soon as it gets tagged with a ubiquitin tag, it gets like signaled to the rest of the body or the rest of the cell, I need to degrade this. So once again, go back to that video, you'll see proteasome degradation within that video. Really cool to see. So you can imagine that from a drug targeting perspective, imagine you want to get rid of things that are happening that are, you don't want those peptides or proteins around. Imagine just being able to target them, put that tag on your body, does what it needs to do. So IRAK4 is a protein involved here in the TLR pathway. that where they did just this. So they were looking at how to basically lower IRAC. So the next two slides are possibly some of them. Well, I looked at it as quite funny, but maybe marked or not. Acronyms. So this is SAD, which is a single ascending dose arm of the study. So basically, they just said, if we take different doses, give you one dose, what is the size of that impact, right? So this is the placebo effect here, and you'll see that after that one dose, there's a very strong drop off, and then slowly IRAC4 comes back again, right? But very clear that dose dependent, strong reduction in IRAC4. Then we go into MAD, which is the second one I found particularly funny, which is multiple ascending doses. So the concept here is that if we give you the drug, cool, it works. But if we continue giving the drug, does it continue to work, right? So in this one, you can see that they gave the drugs for 14 days and then looked at how things are going. So you can see during the period of time that they have the drug, this drops off dramatically. So this is the paper that details all of this. This program was sold to Sanofi after a successful phase 1 trial. That was large parts of that made that successful was all based on. And this is the publication in Nature if you'd like to go and check it out. So there are a number of additional examples. This was an interesting study on diet. It's called Diogenes. It's diet, impact of diet in obesity. And originally was the Diogenes was a genetic-based study. They then went and did an analysis, proteomics on this as well. They were able to analyze 1,500 samples in under a month. They were able to pull out different types of phenotypes of what was going on with all of this. Really, really interesting study that happened. Once again, I would urge you to take a look and see. I'm trying to give you examples of where, in a clinically relevant way, where proteomics has been applied to sort of study whatever your research question is. Another paper that came out in Cell Systems. So this was a high throughput way of trying to. So we were able to determine who had COVID, right? But there were obviously a spectrum of people. Some people got long COVID. Some people were hospitalized. Some people you didn't even know they were asymptomatic. So for various reasons, it would have been good to know who would fall where, right? You'd be able to manage hospital bed loads. You'd be able to do all these kinds of things. So what this study focused on was they took people with varying degrees after the fact. So they sampled them longitudinally, and then once they were classified, they knew where they ended up on the scale. They then processed them through these high throughput processing mechanisms, which I showed you. 5 minutes a sample in terms of analysis time. They put it through a new software package, and they found 27 protein biomarkers. And using this 27 panel, they were able to predict, at the start, they were able to really accurately predict whether you're going to be a severe case, a mild case, and anywhere in between. Don't know if anybody's read this paper in the room. But a PPCM cohort from 6 countries, am I right? Yeah. And what it was able to do, once again, going through these high throughput processing methods, was then able to develop a panel that had really good ROC-kerg, so sensitivity, specificity, to be able to then try and stratify markers for risk. Obviously, this needs to undergo validation and all those kinds of things, but a very close to home example of where this has been applied. And this doesn't just happen in research, right? Like, we make these things and they get turned into commercial entities, right? So This was a study, I think, that was done in, I have to go back and read it. I think it was about 2013. Cohort of about 5,000 women looking at risk markers for preterm birth. It's biological markers, I think, that get used currently, as opposed to any sort of molecular markers. But based on a 3-protein signal, 3 proteins in your serum, there is an 80% improvement in predictive capacity on when preterm births will occur, who is a candidate for preterm birth or not. And this is a commercial company that's operating out of New York. Aturos is another company based in Ireland. They've got products for being able to determine rheumatoid arthritis from psoriatic arthritis, for determining whether you have organ-confined prostate cancer versus non-organ confined prostate cancer. That is, so often people die with prostate cancer, not of prostate cancer. But what seems to be biologically relevant, or in terms of the pathology of progression, is whether it's organ confined or not. And when the moment becomes it's not organ confined, treatment becomes actually better. And so knowing when that happens helps you with that arm. And then something for associated with diabetic kidney disease. So it's not just about research where you are right here, right now, but you should be thinking about where could we go with this? How could we do something with this, right? And what are the steps that we need to follow to be able to get there? So from our side, I can tell you that we've, at Decipher, you'll see some of us in the room, Hisham, Mutshidzi, and Michelle. What we do would not be possible without them. They really make everything tick over. To give you an indication, every single box here represents 96 samples. So that works out to just under 3,000 samples that were analyzed in the last 6 months on just this machine alone. And once again, going to how do we make this a high throughput ecosystem to be able to do all of this? You can follow us on LinkedIn, you can follow us on YouTube, our website, or you can email me if you've got any questions. This is a link tree to Diplomix. So if you want to find out more about any of these programs that are running there or anything else, Diplomix is heavily involved with training initiatives as well. And there's always something on the go. So also really worthwhile following them. And then obviously the CPGR and IDM, who sort of understood that there was a lot to be gained from putting these two entities together. So yeah, I'll leave it at that, and I'm happy to field any questions.
Thank you very much for a really useful overview and what your owners can do for. Are there any specific questions or comments? I mean,
yeah, thank you so much. In your one of the paper you showed where they looked at the different protein and the phosphorylation of the protein. I was thinking actually, yes, it's very important to sort of look at the protein level, then the activation of the protein. Now I was thinking another tool that how it could also benefit would be in the dynamic of the cells. There's not just the phosphorylation, but there's also the translocation from, for example, from cytoplasm to the nucleus, from cytoplasm to the mitochondria. So is there something that could be looked at?
It is looked at. So I didn't go into it yet, but spatial proteomics is a massive, massive thing, because you can have things that effectively don't really change in terms of, so you see them, if you just look at the cell indiscriminately, this cell to this cell, and you just pull out a number that says, oh, it's there at this amount and it's there at the same amount. But fundamentally, it goes from location A here to location B there, and that has an impact. So this is done. There's ways and means of being able to, using ultracentrifugation and other methods, to be able to kind of have a way to capture what's happening across the cell, different organelles and things like that. And then there's also, I didn't talk about it much here, but imaging-based proteomics, right? So many of you might be familiar with pathology and H&E staining, right? You're looking at two markers. You can stain with those two things, they will stain something very particular. That information is given to you on two levels. Okay, if you have to do imaging-based mass spectrometry, and you can either choose lipids or proteomics, you're now getting hundreds of markers that are coming through. So if you had to think about this from a FACS perspective, I think the best FACS facility machine right now downstairs, I think it's a 48 laser or 48 channels, right? It's still only 48 markers, not hundreds of markers. So the depth of information you can acquire in a spatial manner it definitely does exist.
There's just a few comments from my side. I mean, we obviously did the project with you. We continued analyzing the data. So this was a study in part on cardiopsy and led by Taras. Also, maybe Taras is now senior scientist, my last PhD is a senior scientist now in Harare and building his own lab. I'm going to visit him in September. So for him, this paper was very important because he got a permanent job. So that's very important from that perspective. But he also analyzed the data more, and what is important, there was no difference in a certain protein upregulation according to ethnic groups. So it's really seemed to be linked to disease and not in ethnicity or how people present. And it also had an impact. I mean, we found, so it's not just that you measure something. I mean, we found really the coagulation pathway proteins are regulated. And we knew for a long time that women with this type of cardiomyopathy are particularly prone to get left ventricular clots. We never really paid that much attention. Everyone paid more attention to the heart failure, the weakening heart. But when we went back to large data set of more than a couple of 100 people, we actually see that about 1/3 dies suddenly. And then we went deeper into more specific data sets, and we found that it was mainly linked to thrombotic events, pulmonary embolus and strokes. So it really made us aware of perhaps another pathway which is supported, not just weakening the heart, which is bad enough, but obviously the other aspects, how it can also die and it could lead to therapy. So I think it was very useful to process relatively small cohort, but I think the findings are quite important.
And that's such a beautiful example, right? Because show me an immunologist and I'll show you what their favorite cytokine might be. Because typically that's immunologists are like, I'm focused on this or that, you know, IL-10 or whatever the case may be. But there is, you know, I try to get across to you the complexity of what's happening at any given point in time within a cell, within a, you know, that then is part of a tissue, an organ, a body system, all of these things happening concurrently, right? And when you only focus on one little tiny dot aspect within it, you can miss these kinds of connections. And it's by being able to look at these in a big helicopter view where you're kind of unbiased, it's hypothesis generating. You're saying, we don't know what's going on, but we know that something is going on. Let's like get the data and then take a look at it and try and develop ideas around what this is and then validate that.
I have one last question to you. But I mean, I think it's important for, especially for students, so you can measure it, but then you still need to interpret it. The statistic is quite something.
Yeah.
So perhaps you can add or do recommendation what kind of training students should look for, or I know that I obviously looked at your website, but perhaps you can quickly summarize for those who haven't looked at it yet.
So what I can tell you, so stats is always, I hold them with a pinch of salt. There's lies, damn lies, and statistics, right? You can manipulate data however you want to get the right stats. I wouldn't suggest you go about doing that, but you've got to understand Like, what is it that the statistical test is trying to achieve, and are you applying it in the right way? One of the things I get asked most commonly is how many samples should we use for this? You know, doing power calculations and things like that is not necessarily easy. There are resources that exist, but fundamentally, what I would suggest, sorry, I'm chopping and changing here, but there is a biostats department based in Falmouth building. Reach out to them, have conversations with them. It's a great facility to be able to do that. We have something that we run every year called the Ubuntu Proteomics Summer School. This is a national program through Diplomics. We be out some of the guys who are writing the code that for the tools that are being used for stats packages, who can, you know, come out and explain, you know, what these statistics things are all about. Community, you can comment on that and whether or not you found that useful. So
Yes, it was useful. Especially in terms of the statistical analysis after you get your proteomics. And I think that's where most students are involved, especially with South and chemical. So, and then you realize, oh, it's good to learn R, because this is where you will use the stuff.
And there are other ways to do it, right? Like, so Perseus is another, you don't have to learn R, you can, and there's tutorials online for Perseus. That's one of them. Mass Dynamics is one of the tools that we're wanting to bring on board. As soon as we get the credit card payment done, we'll have it on board. So, which allows you to do statistical based analyses on things. Put through your volcano plots, your heat maps, your hierarchical clustering, t-tests, ANOVA plots, all of those kinds of things in a manner that's, you don't necessarily have to do R. So the way I look at it is, some people are going to be really, really interested in the data. And for those people, like, and you're going to become a big data individual, R is probably a good thing for you to learn, R, Python, etc., etc. Some of you are going to be interested in developing a story, more biological context and relevance out of that. And maybe there it's about either finding the right people to work with or finding tools that can do the majority of what you need, and understanding what are do's and don'ts there. And those two pathways are very, very different.
Yeah, how did you get your course for your proteomic data? Yourself? I mean, Vitalis child, they looked online, there were other papers, none of them did the course.
Yeah.
Published, obviously. So, but what did you do?
So fortunately enough, we had a joint collaboration with the University of Manchester under the GP Foundation, and I was fortunate enough to go for training at the Crossway in there.
Yeah.
So that's when we done the SWOT in this. And then the bands, which actually told me and trained me the machine learning algorithms from how we take from the digitized SWOT maps into actual data processing in order to generate those three fields. Yeah.
I think for those
That's a nice resource.
research methodology for us, like what is the future, and you need to understand statistics. I don't think you can be a researcher or biological researcher anymore without great statistics. I don't know how this is going to work. So that's why I'm asking, because you don't have change, because once upon a time, look at what I would my impact. You know, you spend your time in the lab and you pre-fitted your probes and get the results and you put them in Excel. And the main part was the measurement.
Yes.
And the statistics was complicated for a few markers. But now that it's changed, the machines, as you rightly well explained very well, I mean, it's so much superior to us doing certain individual measurements. That will take over, but you still have to. You need to understand what to do with the data. So I think the focus of the training should be on that part. Also, the other part will be done for you. So Yes,
Tim.
hello people online. Is there anyone who has a question?
Sure.
They'll only do online. Oh, okay. No question. No. Any other question or comments? No. Okay, then thank you very much.
No problem.
So for the answers of the key.
Yes, yes.
Thank you.
I should just mention, I'm literally based in the building outside there, that new one that was built that made a lot of noise. I'm sorry about that. But that's where we're based. So if anybody wants to pop across and see everything, we'll just have a conversation one day, you're more than welcome to.
Thanks.
No problem.
I wasn't there when I was in the bank, but I think, Camille,